Hello and good day to everyone. We are Group 1 here to present artificial substitutes to various human tissues. Tissues are groups of cells with similar functions. These groups of cells work together to perform a specific task in the body. The primary types of tissues can be categorized as epithelial, connective, muscular, and nervous. When we sustain injuries, our tissues repair and rebuild themselves. Tissues can be damaged either through acute injuries or overuse injuries. Incidents such as fires can damage these groups of tissues as well. Fortunately, modern medicine has led us to the point where we can provide artificial substitutes for incidents like this. Different tissues need different alternatives.
Epithelial tissue can be found in our different organ systems, such as our digestive system and integumentary system. It exists in various forms, such as simple, stratified, and pseudo-stratified, each with their own subcategories. The need for artificial tissue arises from extreme damages to this tissue through fires or even through sores. Epidermal grafts, specifically, are often made of biomaterial-based matrix without any cells incorporated or are called acellular grafts. They act as a framework for fibroblasts and endothelial cells migration and infiltration upon graft transplantation into the living organism.
Dermograft, on the other hand, or cellular dermal graft, is used for the treatment of burns and chronic wounds, including chronic diabetic foot ulcers. Composed of human neonatal fibroblasts, this graft is plated onto a bioabsorbable polyglactin mesh scaffold. It carries low risk of rejection, but due to its weak ECM structure, it may cause some complications such as infections.
While artificial dermoepidermal grafts are considered as the most advanced type of artificial skin graft available as of late, it is often prepared by a culture of keratinocytes on the surface of the dermal layer with or without incorporated fibroblasts. Let us now move forward to the artificial alternatives for the muscle tissues, as there are currently none that is available for the nervous system.
For muscle tissues, biomedical engineers have used polymer materials, proteins, and stem cells to develop artificial muscle tissues. Polymer artificial muscles are used to develop biomedical devices, such as prosthetics to mimic the natural and efficient motion of human muscles. Some polymer materials that have been used to create artificial muscle tissues have been polymer fiber and nylon. First, polymer fiber-based designs. To mimic muscle tissues, polymer was used to create fast and lightweight artificial muscles. Three different research teams have developed artificial muscles using different polymer fiber-based designs.
A team from Massachusetts Institute of Technology has created a two-phase polymer fiber that can be activated by heat and can lift more than 650 times its own weight while withstanding strains of more than 1000% over thousands of use cycles. This material and technology can be used to create artificial muscles and improve prosthetic limbs. Next, another team from University of Texas at Dallas has created a fiber from polymer yarns that can generate 40 times more mechanical energy per second than human muscle can. Lastly, a team from the University of Purdue created an untethered high-energy microengine composed of shape memory nanocomposite fibers. These fibers are twisted to store mechanical energy that can be released on demand by applying a small temperature change. The devices made from shape memory fibers can be used in microrobotics and small medical devices.
Now on to the second polymer material used to create artificial muscles, nylon. Nylon was carefully shaped and modified and then selectively heated on one side to create a bending motion similar to that of a muscle. Researchers have demonstrated that the material can maintain its performance after at least 100,000 bending cycles and can bend and retract at the speed of at least 17 cycles per second. This nylon system could be used for self-adjusting catheters and other biomedical devices.
Now let us move on to proteins. Researchers have developed artificial proteins that mimic the elastic and mechanical properties of chitin. Titin is a muscle protein in humans and is important in the contraction of the skeletal and cardiac muscle. Researchers from the University of British Columbia and Virginia Polytechnic Institute and State University have designed biomaterials with globular proteins. The resulting elastic material recovers well at low strain and shows great toughness at high strain. Due to these characteristics, this biomaterial can be used in tissue engineering.
Aside from polymer materials and proteins, researchers and biomedical engineers use stem cells to create and grow muscle tissue. Biomedical engineers have grown human skeletal tissues from induced pluripotent stem cells that contract in response to external stimuli, similar to native muscle tissue. The pluripotent stem cell-derived muscle fibers develop reservoirs of satellite-like cells that are necessary for normal adult muscles to repair damage.
So, why might artificial blood or skin be useful? What characteristics would these substitutes need in order to function well in the body? Blood transfusions are one of the most frequently performed operations on hospitalized patients. Nonetheless, despite all precautions taken to protect the blood supply, there are recognized dangers connected with transfusions, including infectious and non infectious problems. Meanwhile, concerns with blood product supply, the necessity for compatibility testing, and blood product storage and shipping regulations have caused obstacles for blood transfusion delivery. There are also those who refuse blood transfusions, for example, Jehovah's Witnesses. As a result, alternative agents that can consistently and safely substitute blood are required. Despite a long history of interest in generating a blood replacement, the creation of effective oxygen therapeutic agents or OTA have yet to bear fruit. This failure may be due to toxicities associated with medicines in development as well as physiological subtleties of O2 supply to tissues. However, the future holds hope, since novel agents, notably hemoglobin-based agents, are now under development, and one agent has been designated as an orphan medication by the FDA for the treatment of sickle cell disease.
Why do real tissues work better? Why not use the real tissues if they work better? Real and natural tissues do work better compared to artificial tissues as they develop from actual cells. They're able to absorb nutrients properly and participate in metabolic activities. Using real tissue from the patient themselves would also reduce immunosuppression in the body due to the biological gene expression. Although real tissues would work better, transplanting or transferring them to the body have some challenges. Some of these challenges are contamination during the culture or cultivation of tissue, vitrification and acclimatization during the transfer or transplant, and the failure of cultured tissue in being vascularized. Additionally, when the tissue being used for the transplant is from another person or another animal even, there is a higher chance of the transplant being rejected by the body, despite it being a real tissue. Furthermore, tissue engineering, which develops artificial tissues, only has a small part in patient treatment as of right now. Artificial tissues still need development to be reproducible, but they can help in the development of drugs and biomedical devices, as well as research by being used to create models of rare diseases for future studies and individualized healthcare.
Artificial tissues for parts that may not be able to regenerate themselves could be used in the future. Nervous tissue, such as the optic nerve, lack the ability to regenerate themselves. It is made up of millions of small connections despite its size. If a substitute can be developed to repair or replace them, many people, like me who suffer from morning glory syndrome, a condition that affects the optic nerve, can get their sight back. An issue when applying this tissue, however, is that there's currently no FDA-approved treatments for the optic nerve, whether surgical or not.
And that is it for our presentation on artificial substitutes for various human tissues. We hope you learned something and thank you for listening.